Okay, welcome back to Module 8. And this week we're going to be talking about molecular biology. We're finishing up the bottom of our triangle and we're talking about reproduction. But not sexual reproduction. We're actually once again finishing our triangle that we've been focusing on this entire quarter so far. So we're focusing on getting this bottom part right here. We've talked about the biochemistry, specifically proteins, those favorites of biochemists. And we've talked about genetics. We've talked about studying heredity and traits and how over time we said that those were carried on genes, right? And these are two different, not the only two, but two different ways of studying biological function, our two favorite for right now. I'm sure that you guys can completely do this by heart at this point right now. But now we're finally getting to this bottom part, talking about molecular biology. We're going to talk about these genes that are carried on DNA are actually encode proteins. But remember, part of this also is that cells need to be able to replicate. So we're talking about, once again, cellular reproduction at this point. So when we looked at this DNA needs to encode proteins, there's another element here. This DNA also has to be able to replicate to carry these traits from parent cell to daughter cell. And that's what we're going to be talking about this week. We're going to be talking about how we replicate cells in a process called mitosis. And we're also going to be talking about making of those sex cells, those gametes, for meiosis. So if I'm going to summarize this week, is these two words right here: replication of cells, either replication of cells for the vast majority of cells in the body, or just replicating in order to create sex cells for sexual reproduction. Mitosis and meiosis are the main two goals for this week. So straight out of your book, what are we trying to do this week? We're going to talk about the structure of chromosomes, and we're going to talk about the cell cycle, typical cell structure, which is actually going to include cell replication, mitosis. I'm going to explain what mitosis is used for, and once again, in what cells and the 4 stages. Then we're going to talk about compare and contrast with meiosis. What is it used for and in what cells? Once again, compare and contrast the two. We're going to compare and contrast making of sperm versus making of egg. So that's spermatogenesis versus oogenesis. We're going to talk about when things go wrong, trisomy, mesomy, when we're, once again, talking about chromosomal disorders. We've kind of talked about that a little bit in genetics. We're going to talk about that again here. What most often causes these changes in chromosome number? What are the symptoms associated with changes in the sex chromosomes? And the 4 changes in chromosome structure. So once again, we're going to talk lots about chromosomes and how they replicate. So just as a quick review, since this is about chromosomes, remember that humans have 46 chromosomes that are in 23 pairs within the nucleus. Now, once again, 23 from mom and 23 from dad. So, pairs of chromosomes are often called homologous chromosomes. So, I want you to be able to kind of get some of these terms down. Once again, because we have 23 from mom and 23 from dad, they're going to be matched up in pairs. And once again, those pairs are called homologous chromosomes. Autosomes, and this is important, are the 22 chromosomes that control traits that have nothing to do with gender, nothing to do with sexuality of the individual. And specifically, obviously, we have one pair of genes, sex chromosomes, that actually do control the gender, whether, once again, those are X and Y. Now, body cells, normal body cells, the vast majority of cells in your body have all 46 chromosomes, right? And those are called diploid cells. Now, very important to get this convention down. Okay, so when you have the matching, the full set, one from mom, a set of chromosomes from mom, set of chromosomes from dad, when you have the full set, you are a diploid. And notice you have the 2n number of chromosomes, which for human beings is 46. We have 46. That is our diploid number, notated by 2n chromosomes. 2, why? Because one from mom, one from dad. When we're looking at the sex cells, the sperm and the egg, right? They have the haploid number of cells. Instead of having one pair from each mom and dad, right? They have only one pair, one of the pair, excuse me, so that's the haploid number. So there's only 23 there, and that haploid number is represented by N. So just a couple important distinctions. Now, when we think about chromosomes, we usually think about them looking something like this, and we tend to think of them these little shapes that look condensed and kind of look like little X's here. Now, really, most of the time, they don't look like this. They only coil up and look like this X, actually a very specific stage in mitosis. However, what we can do, just so we can actually look at a person's chromosomes and look at their DNA. Now, remember, virtually every cell in your body is going to have a nucleus with DNA in it. One notable exception would be your red blood cells. By the way, they do start with that way, but they end up losing their nucleus at one point in development. So every cell in your body, with exception of a few notable exceptions, will have the same 46 chromosomes, other than, once again, your sex cells, your red blood cells, so forth. And most of the time, they're kind of all coiled up and they're kind of this jumbled looking mess. However, at a certain point of mitosis, which you will learn about shortly, they coil up and they give you that nice X look right here. This is one chromosome. And this is during mitosis when they look like this. And notice that, once again, if we stop mitosis at a certain point, and there's ways of doing this, as you might be able to guess at this point, we can actually freeze mitosis and fraction off the chromosomes, separate them out from the rest of the cell, and stain them appropriately. And then we can use a computer to actually match up. This is actually very, very cool. Match up the homologous chromosomes. Okay, so this is an example of a karyotype where we've taken a person's chromosomes and we've stopped it right in the middle of mitosis, and we've stained them, and we've matched up the homologous pair. So here's a homologous pair, okay, and if we look over here, here's another homologous pair. They're all, the computer matched them up. So, for example, let's say we got this one from mom, once again, and this one we got from dad. All right, and there's this pair all the way through. And as you already know, often with traits, there's going to be on these chromosomes, millions and millions of base pairs of DNA, and this is what's encoding your traits, your heredity, what proteins are going to be made, all one and the same, if you will. And there's often going to be a dominant versus recessive trait on these pairs. Which one is going to be dominant for a particular trait? Well, we have to look at each one individually. But we can do this now, which is actually rather spectacular. So we can actually do this karyotype and look at a person's whole set of chromosomes, all 23 from mom, 23 from dad, 22 autosomal pairs, these first 22 along here, and then we finally get right here, there's the sex, that 23rd set, which is the sex chromosomes, And as we can see, this particular karyotype is for a male because it has both an X and a Y. It's the Y that's going to make this one a male. XX female, XY male. So just a short little introduction leading to just kind of a refresher a little bit as far as about chromosomes and DNA, just a little bit, but we're going to jump quite a bit more into it now and talk about, once again, mitosis and meiosis.